Without further ado, our speaker tonight is Chris Anderson, who is an assistant professor of bioengineering at UC Berkeley, my alma mater, go Bears. And he'll be talking tonight about synthetic biology. Welcome, Chris.
Thank you. How are we doing on sound?
I'll turn it up.
All right. How are we now? You hear me in the back? Great. Terrific. So I want to tell you today a little bit about synthetic biology, which is a field I've been involved with for about 10 years, I would say, although people didn't call it synthetic biology before that. I actually came to this field from protein engineering, and protein engineering is one of kind of the three fields that kind of came together to make synthetic biology. The folks in protein engineering were already used to working with the organisms and thinking about problems, but usually we just dealt with a single protein at a time. And then you had a bunch of computer science type folks who came into it, who were looking at it totally differently than the way we saw it. And then you also had these sort of metabolic engineering folks who were already sort of working in this space, but doing it in a much more traditional way. And these three fields have kind of all come together to give rise to synthetic biology, and that got started maybe around 2001 were the first few papers of this field. So in general, I want to tell you about what it means and what are the sorts of things going on here in the Bay Area at Berkeley and UCSF and in companies, and also what's going on in my own lab at UC Berkeley. So first of all, what is it? Now, if you ask a synthetic biology person what this field is about, you'll get a different answer from everyone you ask. It's a little hard to define exactly what this is, and it means different things to different people. And it's kind of changing over time. The sort of things that were being done maybe 3 years ago are pretty much no longer in vogue in the field. It changes pretty quickly. But in general, this is the way I define it. I consider it ground-up genetic engineering. We add DNAs into well-characterized model organisms such as E. coli, yeast, or mammalian cells to elicit new and useful biological functions with applications in things like bioenergy, therapeutics, what else do I have in there? Bioremediation, chemicals, and materials production. And I think still the application space associated with synthetic biology is still something we're trying to figure out. But in general, all these existed beforehand as applied microbiology sorts of fields. And synthetic biology kind of took a new view of all this applied biology and said, all these have a lot in common. The tools that we're using and kind of the obstacles that we run into in making these systems are all very similar. They're very different applications with very different biological basis, but they all have some relationships in terms of the technical foundations. And one of the big ideas in the field is this idea of putting the engineering back in genetic engineering. So genetic engineering it's pretty much been an art form for the past 30 years, but it's been around for 30 years. I mean, it's not a new field at all. But the tools are very nuanced. It takes many years to get into it. If you want to do genetic engineering, traditionally, you had to get a PhD for sure, and you probably still wouldn't be very good at it until you had gone into industry for maybe 10 years and really learned all the nuances. And part of the reason for that is the tools are all very particular and finicky. And also the theoretical basis for why certain things work and how you solve problems wasn't in any way standardized or formalized or well-rooted in some kind of theory. So this idea of putting the engineering back in genetic engineering is to make genetic engineering more like other engineering disciplines like computer science or mechanical engineering, which have just very well-defined tools and standards and approaches to problem solving. So there's this big gap. There's this big kind of spectrum in between science and engineering. We have both. You have biology and chemistry. You usually think of those as science, and then you have the things that are actually called engineering, like mechanical engineering or the building of aircraft. These are engineering disciplines. And the real distinction between these things is, science is traditionally about discovery, characterization, and the theory that evolves from doing that. But generally, it's about characterizing natural systems. So maybe you're studying rocks and you're a geologist. You're trying to understand rocks and the history of Earth or something like that. Whereas in engineering, you're dealing with the design, fabrication, testing for making things. And there's still theory associated with that, but principally what you're concerned with is the theory of how you go about building things. And there's kind of this whole big spot in between where genetic engineering sits that is applied science. And there are a lot of other fields. A lot of chemistry also fits in this void of applied science where you don't necessarily have a very firm conceptual understanding of exactly what's going on in your system. You can't describe it all very simply with differential equations or some other quantitative model. A lot of it is still very intuitive, but you can still make things with a purpose in mind. It's purposeful building of things that are going to be useful to solve societal problems or make you money or whatever. But in general, what we're trying to do is get out of that regime into something that's more like formal engineering. And I would say this aspect of genetic engineering is something that we talk a lot about, and it's something I'm very actively involved in doing in my own lab, but it's something that has not borne fruit yet. Such that today we're still definitely in that applied science space in synthetic biology. But this is where we're going. But in general, the challenge of synthetic biology is how you go from something that you know you want to happen down to genetic code. And in that sense, it's a lot like software engineering. There's a code. We don't have zeros and ones, we have A, T, C's, and G's, but we're still just trying to figure out what is the code that's going to give rise to the behavior that we want. And the basic first idea of synthetic biology is we're not playing with weird organisms. We're not going to go look under roots in Antarctica for the function we want to have the behavior we want that's going to be useful. We don't go looking for biofuel bacteria in Antarctica. We build them based on organisms we already know a lot about. So there's a heavy focus on E. coli work, but also yeast and mammalian cells. And the theory there is if we're all working in the same context, we can compare experiments across different labs. I can say be working in my lab on some device associated with tumor-killing bacteria, where someone on MIT is working on some toy system that's some transcriptional circuit, and we might not know anything about each other's efforts. But if we're both working in the same organism, our two devices can be later reused in the same context, and we have some predictability that those devices are going to function in our own labs, which you can't do if you're playing with kind of obscure natural isolates. So traditionally, genetic engineering has focused on single gene systems. So this has been largely the paradigm for the past 30 years, and it's been very successful. So this is sort of the therapeutic protein paradigm here, where you would start with a gene that you've identified from some natural organism that's going to encode some therapeutic protein, or say insulin, which is a protein that's naturally in your bloodstream, which if you're diabetic you have to inject. That used to be incredibly expensive to get the insulin because it had to come from natural animal sources. But what you do here is you take the DNA that encodes that protein, you use recombinant DNA technology to put it into some sort of cloning vector that you can introduce into some cell line. And often that cell line is E. coli, but it may also be mammalian cells or yeast or Pichia were popular for a while. And then you're going to somehow turn this gene on so that it's going to make lots of protein that then you sell for billions of dollars. And this is the whole basis of the wealth of companies like Genentech in the Bay Area. But there's a big thinking out there, certainly in academia. I don't think it's happened yet in industry. I mean, it hasn't kind of filtered to this state yet, that a lot of the single gene targets that are of significant financial value have already been tapped. And a lot of the applications that will come in the future are not going to be associated with systems that are expressing a single gene, but rather systems that are expressing many genes. So largely that's what synthetic biology is about. So there's this first idea of standardization of the organism, and also this kind of approach that's going to be a little more standardized and methodological in nature. But also there's something qualitatively different about synthetic biology. We're not so much interested in this scale of how we go to things on the size scale of a single gene encoding a single protein. We're more concerned with making larger things. So we're either going from pre-existing proteins to multi-protein devices that are encoding some higher level biological function, with the ultimate goal of being able to make entire genome scale organisms that are doing something fundamentally different from existing natural organisms, but in a very rational way. So first thing, this is kind of an aside, but if you learn nothing else tonight, I hope you will learn this. E. coli is the organism that most of the people in this field work with, and it's a very popular organism for making proteins or small molecules in industry. But probably what you've heard about E. coli is that you find it on your hamburgers at Jack in the Box and it makes you sick. So why are we playing with this organism that makes you sick? So there's a big distinction between these two organisms. The thing you hear about on the news is not actually the same bug that we play with in the lab. So I like to say E. coli are people too. They're good E. colis, they're bad E. colis, they're the Hitlers of the E. coli world, and they're the Gandhis as well. They're not literally people, but you get the point. The E. coli that we play with in the lab is based on a commensal strain of E. coli. So if you were to swab someone's intestines and ask what are the bacteria that exist there, something on the order of 2% of the bacteria in your large intestine are going to be E. coli. Of the bacteria that are able to grow in an aerobic environment, meaning there's oxygen, you call this a facultative anaerobe or an aerobic bacterium. E. coli is by far the dominant bacterium that's able to grow in the presence of oxygen. But there are particular isolates of E. coli that are pathogenic. So there's some that even can cause meningitis. It's a very diverse species in terms of the strains and the range of environments that they inhabit. The distinction between the bacteria that we work with in the lab, the commensal strains, and the ones that are pathogenic, that give you diarrhea or worse, is the presence of virulence factors. So these are particular sets of genes. They're extensively studied and well cataloged, and certain strains have them and other strains don't. But in general, you get multiple categories of types of E. coli. Some of them are commensal. They're normal, they're healthy, they help you digest your food, they're good for you. And the particular lineage that we play with in the lab is the MG1655 lineage. A more modern version of that is DH10B. So this guy MG1655 is the granddaddy of DH10B, and that's a very popular strain in labs. But you get all these other things. And if you were to PubMed E. coli, so PubMed is like the Google of, it's not really Google, but it's like a literature search engine. You can use it to read about anything in biology. A lot of it is open source now, so you can literally read it without being on some campus that has a subscription. You're mostly going to hear about these other things like enterohemorrhagic. That's the one that is the 0157H7 serotype. That is the one that goes on hamburgers. And then you get all these other categories like enterotoxigenic. So there is a distinction between these organisms. And the one we play with in the lab, you can drink it and it doesn't cause sickness. It doesn't have virulence factors.
You wouldn't recommend drinking it, though.
I wouldn't recommend it, but there's a very famous story. The guy who discovered the genetic code, Sidney Brenner, he drank a flask of it. It was a big to do, just to demonstrate really you can do it. It won't make you sick. All right. Another big concept in the field of synthetic biology, though, is this idea of parts. Parts are defined DNA sequences that encode primitive elements of biological function, such as protein coding sequences, promoter sequences, and ribosome binding sites. So what I've designated here with an arrow is the coding sequence for a protein. The name of the protein is going to be LuxR, and it's going to have some DNA sequence associated with it. And traditionally in genetic engineering, we just draw the arrow, but the arrow didn't have any explicit meaning. It just kind of meant the gene, but it wasn't a particular sequence. It was something that encoded LuxR. A big part of this fundamental idea of putting the engineering back in genetic engineering is this now is going to mean something explicit. The part is going to mean a specific DNA sequence. You can have physical manifestations of that sequence in the presence of DNA, that is the little, drips of liquid we play with. And then within a cell, you can have some physical manifestation in the form of whatever that piece of DNA encodes. But then beyond that, you have devices, and devices are going to be defined as collection of parts that have some human-defined function. So in this case, we have about 12 genes that are going to encode a biosynthetic pathway. So a biosynthetic pathway is going to be a set of genes that encode proteins, and those proteins are enzymes. Enzymes are a class of proteins that do chemical reactions. So in this case, you're going to have 12 genes that each take the product of the previous reaction and kind of convert it into something and pass that product on to the next enzyme. And so a biosynthetic pathway is a string of these enzymes together that takes some input small molecule into some output small molecule. So you can get things that do chemistry like that. You can also have things that are just regulatory devices. So in this case, this is one that's pretty simple, but it's gotten a lot of attention. It's a receiver device. And basically what's going on here is you have transcription factors, which are proteins that are going to affect the activation of genes. And what you can do with this is cool stuff like this. So what you're looking at is little spots of E. coli on an agar dish. And in the middle, you have a sender cell, which is emitting a small molecule. And it's diffusing out. And then each one of these has this receiver device encoded within the bacterium. And basically what they're doing is sensing the signal and turning on. And you can generate really interesting patterns on plates. Why you want to do this, I'm not entirely sure. A lot of the early work in synthetic biology was kind of based on toy systems that were fun and interesting, but not necessarily useful. But it was just, the first phase was just getting people to think a little differently about what you would be doing with DNA in cells. So you can have biochemical devices and you can have regulatory type devices. And a lot of people in synthetic biology wouldn't even include the biochemical type devices as being part of the field. I am not one of those people. I think both are, it's hard to distinguish them, and a lot of the applications turn out to have bits of each. But to explain the difference between these things, consider this device. I think everyone in this room could build this device, possibly has built this device, just a battery and a light bulb. It turns on, it glows, that's it. It's kind of the equivalent to having a gene under a constitutive promoter into a cell and that gene encodes a protein like GFP that makes the cell turn green. So the cells are just green all the time. But you could do kind of a variation on that device where, say, you replace the light bulb with something a little more elaborate. You spend a lot of time designing this neon lamp thing, but it's still just going to be on all the time. But you put a lot of attention into the colors and the manufacture of this particular light bulb. It's a special output to the thing that has special kind of characteristics. In general, we're going to have the equivalent in genetic engineering where you can be manipulating the biochemistry of the system. So it's kind of the who and what that's inside the cell, the kind of chemical composition of the cell. But you can also have regulatory devices. So this is Hal, the computer. In terms of the light bulb aspect of that, he's pretty trivial. He's just a light bulb. But what's interesting about it is the regulation of that light bulb. There's a lot of logic going on behind the scenes, which is just control of the flow of electrons from the battery to the light bulb, but in a very complicated way. So similarly, we can have regulation in biology, where we're not necessarily affecting the who and what of the cell, but just whether it's turning on or not, and that can involve logic, it can involve timing, that sort of thing. So that is what regulation is. So the applications of synthetic biology are pretty broad. The first big thing that put Berkeley on the map in this field and has made UC Berkeley kind of the lead institution in all of synthetic biology. Part of it is we just have a lot of people doing it, but the big thing was this work here. So this was done in Jay Kiesling's lab at UC Berkeley. And it was all about treating malaria. Malaria is bad news, kills a lot of people, not really in the United States, but out there in the rest of the world, it kills a lot of people. 1.5 to 2.7 million every year, a lot of them are children. Bad news. Well, it turns out there is somewhat of a cure for this out there. A lot of the problem with malaria is it's now resistant to some of the first generation drugs, but there are kind of second generation drugs that are known about that can treat malaria, but they're just not really available at the price and quantities that you would need to treat the number of people that have malaria. So this particular small molecule is called Artemisinin. It's derived from a somewhat obscure plant, and it is a very good cure for malaria. Unfortunately, it's in very low quantities within the plant. So what the Kiesling lab people thought would be a good idea is, why don't you take all the genes for this and build up a new biosynthetic pathway in a microbe, like a yeast or E. coli, to make lots of this small molecule really cheap, and then you can give it to everyone in the world. And they were successful in doing this. So the system I showed you earlier, the biosynthetic pathway, took about, I think it's 12 genes, I'm not going to count them, but it's 11 or 12 genes that they put in the end in the yeast is what they ended up going with. But they were able to, through optimization of this system, get the yields from levels that were kind of comparable to the plant up through many orders of magnitude, such that now this has gone into production. And one interesting point about this, I've colored these different genes based on their source, and there are actually 3 different colors, if you can tell. That's the same as that. One big idea in synthetic biology is you really don't care where the genes come from. All you care about is the functions that they encode. So you have 3 different sets of genes, all coming from different organisms. Some of them are coming from the plant that made this small molecule in the first place. Some of these genes are going to come from E. coli, some from yeast, or from anywhere. And it requires that you think about biology a little bit differently, because it no longer is just relevant where the DNA sequence came from. It's just a sequence. It's just A's, T's, C's, and G's. It doesn't really matter that it came from a plant or a human or a bacterium. As long as it has the particular primitive biological function that you care about, you can put it into a new system and encode that function. So what this is all morphed into, oh, go ahead.
So how exchangeable are these?
Here, let me pass you the mic so everyone can hear you.
How exchangeable are these genes or these sequences that can produce these things between different organisms?
So there's a technical aspect to that and a biological aspect to that. The technical aspect of this is you don't even have to physically grab the DNA from the organism. All you have to know is the sequence, and you can use chemical approaches to build that DNA and put in your organism. So you don't necessarily have to have a physical source for the plant or whatever it came from anymore. The second part of that is biological. What is the probability that if you take a given DNA sequences from an organism, that's going to function in your other organism? And in general, if you stay within the same kingdom of life, it's almost 100% chance that it's going to carry over in terms of just the protein's going to express. So that, at the protein level, that's true. If you're talking about regulatory parts, things are much more nuanced. But when you cross kingdoms, it drops down significantly, but it's still something on the order of 20 to 50% probability of success, and it's often predictable. Just there's so much history of people putting one gene into these organisms and telling you whether it worked or not that we kind of know more or less what works and what doesn't. The big thing is biofuels, though. And again, this is a small molecule production problem, but the yields needed to do biofuels means that you have to get the regulation right as well. So it's actually a much more mature sort of problem than these more traditional metabolic engineering applications. But in general, the scheme here is you're going to have plants. Those plants might be genetically engineered, they might just be natural plants that are good at making biomass. You're going to harvest the biomass, and then you're going to use some degradation process, which potentially involves an engineered microbe. It could also be a kind of chemical engineering sort of problem, a chemical process that you're doing to this biomass to get to simple sugar, which then you're going to ferment with this highly engineered microbe to make something other than ethanol. You're going to make something that really you can put in a car and get the same sort of energy density that you would get with gasoline. So really you're making the same molecules that are in gasoline, but you're making them by fermentation with an organism. And this has been huge in our field, and it's huge in the Bay Area. There are a number of companies doing this in addition to the institutions that exist at UC Berkeley for biofuels. So this company, Amyris, they actually got started making Artemisinin. So Artemisinin started in the lab, in the Kiesling lab at Berkeley, and once they had that more or less working, they launched this company, Amyris. It's in Emeryville. And then they finished the project, so that's in production now, so they don't really even work on it anymore. But then they switched gears and started working on biofuels. There's another one, LS9, which is their main competitor. And then there are a few other companies that I know less about, but are also in this space, Solazyme and Codexis. And there are probably a bunch of others that I just don't know about or I forgot about. But my own lab, we don't work on biofuels at all. We principally work on tools and therapeutic bacteria. So my general interests, though, are in the applications and foundational technologies for this field. And there are kind of three projects that everyone works on. It's kind of get broken up into smaller projects, but the three kind of areas are engineering tumor-killing bacteria. So these are anti-cancer therapeutics. Engineering cells to write DNA. And the third is something that I'm not even going to try to explain. It involves genetic selection of non-natural parts. It's another foundational tool. But I'll explain the first two to you. So the idea of tumor-killing bacteria is to make a bacterium that can be safely injected into the bloodstream, will localize to and invade cancer cells within solid tumors, and then kill them. And this actually is a pretty old field. It's just the approach that we're taking to it is new. And it might strike you as a very strange idea to use bacteria as a therapeutic. In general, you think of bacteria as being associated with disease, not associated with therapies. But you can go into the grocery store right now and buy this product, Activia. And people do it all the time, and it contains a live bacterium. And people intentionally infect themselves with this bacterium in the hope that it's going to improve their digestion. And that's actually is pretty popular. I was back in North Carolina last year, and I saw a whole shelf of this stuff, this cheese product, which is kind of the second generation of this stuff. So people really like these things. But also, if you were to be so unfortunate as to acquire bladder cancer, acute bladder cancer, the primary treatment for that is a bacterium called Mycobacterium bovis BCG, which is kind of a traditional genetic engineering derived product. It is not a natural bacterium. It has some added DNA into it, but not that much. But basically what they do with this is they'll catheterize this stuff into your bladder, where it elicits an immune response that results in the destruction of the tumors there. So in general, what we're trying to do with bacteria is engineer something that's a very smart drug. So there's an existing type of drug for cancer that is referred to as a smart liposome. And a liposome is a chemical product. So basically, you take the lipid things that make these bilayer sacs. So it's kind of liquid in the inside, liquid on the outside, and the fatty things surrounding it. It's like a balloon. And you fill the inside of these little balloons with something that's an anti-cancer agent that's really nasty, like a traditional chemotherapy type agent. But then you put on the surface things that allow that thing to stick to the surface of a cancer cell via cancer-associated epitopes. So these are little signals on the presence of cancer cells that make them look different than healthy cells. So this thing can somewhat specifically bind to cancer cells and localize the delivery of the toxic small molecule to them. So the motivation for using bacteria instead of this is in part that it can do all the same sort of things that you can get out of a chemical system, but allows you to do a lot more things. Like one thing that I think is actually quite major in this space is the fact that you can encode the ability of a bacterium to swim. And this has a big impact on the ability of these small things to penetrate within a tumor, which is a major issue with cancer therapies. So the other thing we work on that's major is the ability to write DNA. So, this whole field is about building DNAs, putting them into these model organisms, and then see if they encode the functions that we design. So it ends up being a fairly cookie-cutter process. You have a batch of bacteria, you are making new DNAs each time, but the methodology for making those DNAs is kind of homogeneous. So you're doing the same thing over and over. But we still do it like this. We still do it kind of one at a time. You get these messy gel rigs. You look in our freezer really looks like this. That's not literally our freezer, but it does look like this. You have all these boxes that are hand labeled. It gets very messy. It makes things slow. It reduces the scale at which you're able to do this sort of research. So where the field is heading, and a big part of what we are doing, is trying to automate this sort of research. So the first part of that is automating the DNA synthesis aspect of it. And the second part of it would be trying to automate the analysis aspect of it. And once you're able to do that, you've largely taken the wet component of this science out of the hands of the engineers. It's all being done by machines. So all we do is sit behind our computers and think about what's going on. And there are two robots in particular that are kind of interesting to us. So actually the field of robotics associated with delivering liquids for these sorts of applications is very advanced. And the reason it's advanced is they use them for small molecule screening in industry. It's way more advanced than the tools that we can put into these systems. So one of them is this thing called the Raptor. This is I think that's a 384 well plate, but it can also take 15 36 well plates and 34 56 well plates. So these tiny little volume wells. That whole thing is about this big. I should have brought one. They're cool looking. But these tiny little wells, and it's spinning very controlled amounts of liquid into each well. So it's dispensing, and it has a choice of 8 different things. The other type of robot that's kind of interesting to us is this acoustic liquid handling robot. And basically what this does is cherry pick. So it does well-to-well transfers of liquid. So you have 2 plates that it puts against each other and it moves them so that it aligns the wells, and then it sticks this acoustic probe underneath them that makes the liquid jet out towards the other plate. So you have this ability to do liquid transfers both in a dispensing mode and a cherry picking mode very quickly with no consumable products. So if we can make all of synthetic biology reduced to small volume liquid transfers, we have awesome automation options for scaling this stuff up and driving the price to basically nothing. So that's what we're trying to do with synthesis. And I'm not going to describe this in detail because it would be very complicated to do so. But I'll give you the very big picture idea of this. So first of all, you should know this is very different from the way you do DNA assembly today. Today, DNA assembly is done starting with a chemical process. You have these chemical machines that have the monomer components of DNA in some chemically activated form. And you have machines that are going to do chemical reactions under, water-free conditions on a solid support. And you build up things that are about 100 base pairs in length. And then you can do a in vitro reaction where you're taking those 100 base pair things and assembling them into 1,000 base pair things. You confirm the sequence of that 1,000 base pair thing, and then you can assemble those into longer things. This is a very different approach. Basically, we're starting with 384 individual types of phages. So a phage is like a virus that infects bacteria. And each one of these little phages is going to have a triplet sequence. So you have all possible triplets of things like GCA and GAT. And you have these arrayed in plates. And what you're going to do with them is jet 2 at a time into this highly engineered bacterium that we call the assembler cell. And what the assembler cell is going to do is join these two DNAs housed in the phage. It's going to turn on the program that lyses the assembler cell, releasing the product into the lysate, which is CGAT. So it's going to join that GA to that GA to make the former CGAT. And so you do this over and over, and you can build up sequences by joining 2 at a time. So this is what we're trying to develop. And the goal is just to be able to get this all so we can do it on very tiny volumes and use those robotics that do those small volume liquid transfers. So I think that's the end of my introduction. Do we have any questions?
I will come around and just throw your hand up. I'm going to start in the back. Oh, so neglected back, and we'll work our way for it. We'll start here in the middle, actually. Yep.
Okay. My question is, do you ever get any surprise when you put genes together or parts together? Does it not work because there's some unwanted or unforeseen synergistic effect between the two genes? I mean, how can you tell when it's a surprise synergistic effect and when it's because it just doesn't work?
So just doesn't work is extremely common in this field. And just doesn't work for reasons we don't know why it just doesn't work is extremely common. If you just make a sequence that is, say, a constitutive promoter and the coding sequence of a protein and all the other accoutrements you need to make a functional gene, like a ribosome binding site and terminator, you just put that together, the probability that that thing will just not express, like it doesn't turn on, is about 50%. So it's kind of like you hooked your battery up to your light bulb, and half the time it just doesn't turn on for no good reason. I mean, the connections aren't bad, there's no mutation, and it just doesn't turn on. And figuring out exactly why that happens, or figuring out a standardized workaround for that problem, is probably one of the biggest things that will increase the pace of this research. The other major unknown is, we're principally dealing with regulation type engineering. And in regulation, you always care a lot about the particular expression level of the system. When you're doing just biochemical engineering, you just want more is better. More protein, more therapeutic protein you can make in the cell, that's all you want, that's all you care about. But when you're talking about regulation, it's you want the right amount of protein, which might be a fairly small amount. And being able to set genetically the amount of protein product that's coming out of a gene, we have no ability to control that yet. So these are the major issues. Now, do you tend to get biochemical functions that were not predicted? No, you don't really get that. I mean, if you put the gene for GFP into a cell, it turns green, it never turns red. It's, in terms of the composition of the things that you're getting out, that's pretty routine.
To kind of build on that question just really quickly, how did you decide that you wanted to use CGA and GAT in that example that's on the screen right there? Is that somebody else's lab that comes to that conclusion and then you're just building off somebody's work?
So in this case, we have to be able to do every single type of pairing of 3mers. So we've made all possible 3mers. So that's n times n times n, or 4 times 4 times 4, which is, I think, 64. had to do that. I don't know what math is.
You can't do anything without your computer anymore,
I
can
can't.
you,
I need
Chris?
Excel for this. I can't do 4 times 4 times 4. It's too big. I don't have enough fingers for that. But we have to be able to do all the permutations to be able to make up any arbitrary DNA sequence. Now, how is it that you know what the DNA sequence is you want to put in? Well, in this field right now, we're always starting with known DNA sequences. So we're not necessarily making up new protein functions. That's really the arena of protein engineering. Largely what we're concerned with is assembling proteins together in a way that they're going to do some higher order function. So if we want the gene for GFP to be encoded in the DNA, we look on PubMed for the sequence of GFP, and we build that sequence into our circuit.
I went to a talk in which a speaker said that you could actually purchase DNA synthesis machines on eBay. Are the machines that people buying the ones that you showed up there? And another question is how accessible in general is playing around with this stuff to the public at large? What kinds of things are people doing?
That's a great.
Just before you answer that, so are you in the market for one of these machines? Are you looking for a recommendation?
Preferred models.
No, but to go back to this topic, can you answer first? Like, do you know, is there a public interest in developing this kind of work outside of the realm of academic research?
Yeah.
Oh, wait, there's a
These
comment
are
right
very different questions. Let me start with the first one associated with DNA synthesizer. So a DNA synthesizer is the thing that makes oligos. In oligonucleotide chemistry, or phosphoramidite chemistry, it's also referred as, gets you up to about 100 base pair length of single-stranded DNA. So you can't directly put that into a cell. First of all, it's nowhere near big enough to encode anything. A typical gene is on the order of 1,000 base pairs. And when you're talking about these device-like systems, you know, if you have 4 genes, then you have something on the order of 4,000 base pairs. So the oligos that you get out of a DNA synthesizer are themselves not enough to do anything. But they're in vitro reactions that you can use to assemble those oligos into larger things. And there is no machine per se that is referred to as a DNA maker or something that does anything other than what the DNA synthesizer does. So if you look on eBay, you can get a DNA synthesizer that'll make oligos, and in general those ones that you would get on eBay are the traditional type that would do about 2 to 8 oligos at a time. So you can do 2 to 8 oligos, so you get about 200 base pairs to 800 base pairs in one run. Today, no one has those machines anymore in academia or in industry because they've been replaced by multiplex DNA synthesizers that are able to do things many more oligos at a time. And the people that have those are the companies. So it's no longer cost effective to run a machine like that to do this kind of research. So there is no machine. The robots I was telling you about are generic liquid handling instruments that are just for moving any kind of liquid. They're principally designed to move solutions containing drug-like molecules, not for doing this. So we're kind of repurposing a set of robotics for these kind of applications. The second question was about people doing this sort of research outside the context of a traditional academic lab or an industry lab or a government lab. And there are a lot of things underway to do this kind of open source, do-it-yourself biology. It's been in the news quite a bit, first coming out of Boston, and there was a recent article about some folks doing it here in the Bay Area. And it's very controversial in the field. There are definitely laws and regulations associated with the handling of these types of materials and doing this kind of research. So doing it in your closet is actually legal, but not necessarily unsafe. So it's very controversial as to whether people should be doing that at all, and trying to come up with a format in which people out there in the world can do this research, but in a legal, safe setting, is something that a lot of people are trying to figure out how to do.
But before we take the next question, does that answer your question? And what were people trying to do in the Bay Area in their closet?
Well, the one thing I read about was what most people end up doing is they have fun transforming GFP genes into E. coli and seeing them glow, which, you know, we don't do that in there's no
So it's not informing the science, it's kind of parlor tricks in a way.
It would be very hard to do it on your own. Things are still really expensive, and there's still a lot of technique to doing this, such that if someone doesn't train you on how to do it, you probably couldn't just figure it out on your own by reading things. But one day it probably will be possible to do this, especially if gene synthesis prices come down to the point where we get rid of the kind of ad hoc procedures that we would currently use to do the DNA manipulations.
Yeah, I think there was a Science Friday a month or two ago. That's probably the one you listened to too, where they talked about these clubs that were doing this and trying to promote it on the level of sort of robotics clubs, even in high schools, which a little scary. Don't get any ideas, kids. My question is, so what you're doing that is different from, say, what some company might do to make, say, a special wood that is water resistant, or an apple that lasts 12 years on the shelf, is regulatory. It's not just finding the particular.
It's multi-gene or regulatory.
Okay.
I mean, those haven't really filtered into the industrial space outside of kind of the metabolic engineering stuff yet.
And you know how to trigger a particular thing, but you don't know how to stop it, you're saying?
No, it's just control. So, for example, if you take GFP and you put a constitutive promoter, constitutive just means on all the time, then you get on all the time. But you can also take a promoter that, say, only turns on at 30 degrees in the presence of melamine. Melamine's that thing that was in all the Chinese dog food or something. And you put that now in front of GFP, and you've got an organism now that's only going to turn green at 30 degrees and in the context of GFP. And when you take it back out of those conditions, it's going to turn off again.
So that's what you're doing.
It's that sort of thing. That's an example of regulation. Not all the examples are like that. That's an example of environmental restriction of the expression of a gene.
Yeah, hi. Right there. Hi. I was wondering what role, if any, the discovery of epigenetics has had on your field.
So in general, in prokaryotes,
Hang on just one second. What's epigenetics for the rest of the crowd? So,
He could probably give a better definition than I could.
but this is a discussion forum, so you're the expert as well.
I can define it for you.
Why don't you talk about just really briefly, like what sort of prompts you to ask that question about epigenetics?
Because it's more than just A, T, C, and G. It's these extra molecules that wind around the DNA helix.
So you're talking. Your question is based on the fact there's a whole system at play, not just the ATCG.
Yeah, there's more.
Right. So in general, the definition of epigenetics is pretty much what he said. It's just it's information that is housed within an organism that is not encoded within the DNA explicitly. Now, sometimes it is indirectly encoded within the DNA in the sense that the DNA might encode the genes that do modifications to the cell that then self-perpetuate. So you can't have stuff like that, but in general, when people say epigenetics, they mean some information that's somehow stored in a state that perpetuates through cell division, but is itself not at all encoded with the DNA, such that if you, in theory, if you made some synthetic cell with just the DNA from that organism, you wouldn't retain that information that was epigenetic. And it's huge in eukaryotic biology. So eukaryotes are us. We're eukaryotes, yeast are eukaryotes, bacteria are prokaryotes. So there are three major kingdoms, right? And eukaryotes do a lot of epigenetics. And pretty much all of what's associated with development, so how you go from a single zygote to a person, the thing that's different about my skin cell from my brain cell, they're genetically the same. The difference is just kind of these stored states. And mechanistically what's going on is you can have genetic circuits where certain transcription factors inhibit the expression of other transcription factors and leave only themselves on, and that can maintain a state that self-perpetuates. You can get methylation of the DNA, so chemical modifications of the DNA that aren't changing the A, T, C, and G, but add little groups to it. There are histone modifications. The big thing of late has been the discovery that RNAs, little tiny RNAs, play a huge role in eukaryotic biology. In general, though, this is pretty much a eukaryotic thing. Prokaryotes don't do it that much. And I think that's a big reason why a lot of us are focusing right now on prokaryotes, just because the range of regulatory functions involved is much simpler. But there are some people in the field who play with epigenetics, such as siRNA, if that buzzword means anything to you, that engineer siRNA type epigenetics to program new biological behavior within a cell. And it's something that certainly when you get into the eukaryotic realm, if you ever want to do anything sophisticated in that space, you're going to have to learn how to engineer that stuff too.
I wonder if you could briefly explain to us how the liposome or other therapeutically engineered entities know that to attack just the cancer cell and not the benign cell. Is it something in the surface of the cell that attracts it?
Did everyone hear that? Could you hear in the back? So the question was, how do you engineer the liposome just to get at the cancer?
For a liposome or for a bacterium?
In general.
In general.
In general. So there's several things that you get. So first of all, you get kind of physical characteristics, very macro scale differences between a tumor and the rest of your body. There's difference in the degree of permeability of the vasculature that are distinctly different, that affect the transport of drug-like things within tumors versus other places. You also get very distinct differences in the degree of phagocyte invasion into tumors versus other tissues. So actually, bacteria, the reason it's an existing field is you can take many different natural isolates of bacteria. You have to grab the right ones. The particular strains have to be the exact right ones, and the organism you choose has to be the exact right one. But you very often get a spontaneous localization effect when you inject these bacteria into an organism that has a tumor. And what's going on is, first of all, the bacteria don't get out of the bloodstream everywhere. And they kind of somewhat selectively get out within the solid tumors due to the differences in the vasculature. But also you get a function where you have differential clearance and growth rates, where either the bacteria just don't get cleared out of the tumor, or they have increased nutrients there that allow them to grow. So that all you get for free. Now, how do you engineer something to be more specific for a tumor? Well, there's several things you can take advantage of. First of all, you have chemical microenvironment cues, such as the concentration of oxygen and glucose. In many ways, a tumor is sort of like a tired muscle in terms of the chemical composition of its local environment. You can also have cancer-associated epitopes that you can use for binding. So these are small things, often they're sugar modifications that are distinctly different chemically on the surface of a cancer cell from other areas. And these are all external to the cancer cell, but then you get a number of things internally that are different between a cancer cell and a healthy cell, such as you pretty much always have two kind of branches of apoptosis pathways that are blocked within a tumor in some way. The P53 pathway and the RB pathway are ubiquitously in some way disrupted, and that could be a cue that you use to gain selectivity.
So let's kind of dumb that down a little bit.
Okay.
Just for my sake, let's just say. Is cancer? All cancers created?
No.
So these are going to have to be targeted for specific cancers. So like if you were attacking a lung cancer,
It can be, okay, so you get some things that are
Is there
kind of
overlap?
universal to solid tumors. But when we say cancer, we usually also are including things like leukemia. And leukemia is a single cell disease. There's no big ball of cells associated with a leukemia. There's single cells floating around. So in that case, you lose all the microenvironment cues, you lose this kind of vascular difference type stuff. But if we restrict ourselves to solid tumors, there's some characteristics such as the low concentration of oxygen that are kind of universal to solid tumors. But you also get things that are specific to particular cancers. So, for example, the HER2 gene, which is the drug Herceptin from Genentech, for breast cancer is directed towards the HER2 receptor. And that is a particular cell surface display cancer-associated epitope that is not present in all tumors. It's more than just breast cancer, but it's not all tumors. So you can have some things that are general, like the chemotherapeutic drugs that were the 70s sort of drugs for cancer. Those things are general anti-solid tumor or anti-cancer. They're even broader than solid tumor because they just affect rapidly growing cells. So those can be very general, but you can also have drugs that are very specific.
So there is some overlap, but not. It is going to be somewhat specific based on the
I think
condition.
in the end, you'd probably try to make therapeutics that were specific.
And just a real quick follow-up again. And is your lab, your research, at any point ready to inject somebody right now?
No.
So I want to get that across because I think that's an important kind of
Don't call me
concept.
asking for bacteria for your grandmother.
No, I'm not asking. I'm definitely not asking for any right now. But just so we get the idea of the scale of where we are in terms of the science right now, where would you say? Are we
I mean, there are
infancy?
people in the therapeutic bacteria space who aren't doing it the way we're doing it, but are using, much less engineered bacteria that have taken things into human trials. And that TICE thing, that's been around for quite a while and is a real approved drug that is truly in use in hospitals. But the sort of approach we're taking is very different, and there's a lot of upfront groundwork that we have to do before we can even make it work in a mouse.
Okay, binding. How many years are we talking about? We're not-
More than one.
You're only saying that because there's video. Video evidence. You don't want to be
I'm
held
supposed to say is the typical lifetime from inception to full development is about 20 years in the therapeutic space.
Oh, that's good. And you've been a professor for 2 years. Okay.
It's a countdown.
Thank you. My name's Narayan Devira. I'm a retired medical doctor, and I learned a lot from your presentation. If I use Kishore's vocabulary, I've been injected. Although it was a drink, I drank a modified acidophilus that produces telomerase enzyme.
Okay.
And so my questions, I think, will interest you and the audience as well. I took this drink, I think, before these new laws came into effect. The guy did the genetic modification in my closet, so to speak.
Okay.
And I watched the whole process, but I couldn't reproduce it.
Okay.
He claims to have patented the genetic print of his modification, so if anybody cheats on him, he can sue them for violating his patent. And I'll just let you comment generally on what I'm presenting, because my personal question is, could this gene be isolated from the acidophilus bacteria in my intestines right now?
Real quick follow-up, Narayan. Do you mind sharing why you took this?
Oh no, a friend of mine wanted to be a client of this fellow, and I got my drink free.
Oh, so it wasn't for a particular condition or anything that you...
Correct.
Okay.
So, with normal commensal bacteria, the maximum sort of turnaround time before they just naturally flow through is, I think it's like a week and a half is max. Without replenishing the supply, things just turn over fairly quickly. That's certainly true of even the best colonizer bacteria. So it's probably not in there anymore.
I'll give you the file.
Okay, thank you very much. He recommended taking penicillin with the drink because his engineering organisms were penicillin resistant, so that would allow greater penetration. But he said he couldn't specifically suggest that because it would be a violation of medical ethics.
All right. Yeah, I mean, that's in that probiotic space. The dynamics of the infection, because you're competing with a whole lot of bacteria that are there, get very complicated.